Interphase, the same thing that's occurred in mitosis is going to occur here. You go through your checkpoints, you decide that this is a cell that needs to become a gamete, so you initiate cell division, your chromosomes have to be duplicated, all those sorts of things have to happen. And again, the cell is performing normal processes until it has duplicated. And now you have your duplicated DNA and you go into prophase 1. In prophase 1, wow, that's a familiar diagram, right? Your centrioles in animals or centrosome regions in everybody are heading for opposite sides of the cell. The nuclear envelope is degrading. Your centromere and kinetochore are forming. Your chromosomes are condensing. But there's an additional thing that's happening. And when we talked about the jumping frogs, we said there is a point where you want information on the two different homologous chromosomes to come together. And to do this, the chromosomes physically pair up, and it's called a bivalent chromosome. So they physically align, and once they become closely associated, they become known as the tetrad. But anyway, the important idea here is the chromosomes, the homologous chromosomes, come together and are physically joined together. And at that point, they align where the DNA is identical. So they find places where the DNA is the same. And then they can physically break the ends of their chromosomes and stick them on the other one. So you now have what's called a non-recombinant chromatid, and this is called a recombinant chromatid because it has recombined. It has broken and changed the DNA. In most organisms, crossing over happens 0, 1, 2, or 3-ish times. It's unlikely that you'll see 25 crossing over events or something like that. But 0, 1, 2, 3, 4, somewhere in there are the number of crossing over events on a chromosome. And after this crossing over event happens, you can certainly cross over again here, or you can cross over with a different chromatid. And why crossover? This comes back to our friendly camouflaged high jumping frogs. At some point, this frog is going to have baby, and hopefully it's going to have a genetic winner where one of the chromosomes will then have both good mutations, and natural selection is all about the winners, right? Frogs have several hundred babies, only 2 of which are likely to survive to adulthood, so you might as well try a whole bunch of times and hopefully you'll get a winner out of it. After prophase is metaphase 1. Be very careful when you're talking about metaphase 1 because people like to say, oh, it's the same as mitosis, and it is not the same as mitosis. In metaphase 1, what aligns are homologous pairs of chromosomes. All of a sudden, your 2 copies of chromosome 1 are associated with each other and your 2 copies of chromosome 2 are associated with each other, then the spindle fibers do attach. You've got your centrioles and the centrosome region here, and that is all still the same, but what is aligned is not one single line of chromosomes along the metaphase plate, but homologous pairs are aligned along the metaphase plate. And these line up in what we call independent assortment. So that means all the chromosomes from mom, or in this case, all the green chromosomes don't necessarily align on the top and all the pink ones on the bottom. You could just as easily have green, pink, or pink, green, or pink, pink. It does it completely randomly, and it doesn't, in this process, doesn't seem to have any idea whether the chromosome originally came from mom or originally came from dad. So the process is entirely independent. And that leads to lots and lots and lots of possibilities, right? Because even in just having a haploid number of 2, a diploid number of 4, when they align, you can align such that the blue ones are on one side and the red ones are on one side. Then you get your haploid cells and they make blue blue or red red. But you can also align it so the blue is on one side of chromosome with chromosome 1 and the blue's on the other side with chromosome 2. So now you've got blue red or red blue, right? You've got 4 different possible kinds of gametes. And if you did 3 sets of chromosomes, you have 8 possible gametes, 2 cubed. Stop the video here and figure out, draw out, think about how many possibilities you would have if you had 4 chromosomes. Once you've got that figured out, then start the video again. Did you come up with 16? And this is where exponential growth matters. If you have 23 chromosomes, that's about 8 million possibilities. And then because any of those eggs can combine with any sperm, that's 70 trillion possibilities. And then there's crossing over, and that becomes essentially an infinite number, or a number not different from infinity based on the total number of kids that any individual woman would ever have. You can make lots and lots and lots of genetic diversity in this way. And somebody's going to say, but Dr. Ward, my brother and I look absolutely identical. And you might. And that may be because a lot of the traits that make you look identical, there's actually not that many genes involved in making those traits. So if you have 4 or 5 or 8 genes that are the same, you may actually look pretty similar. Or it turns out you look pretty similar in some ways, but different in others, and people just focus on the similarities. In any case, there's a lot of diversity, and unless you and your brother are identical twins, you are likely to have many, many different genes from each other, even if some of them are not apparent at the surface. Oh yeah, so plus crossing over, lots. All right, so we've gone through meiosis 1, prophase 1, metaphase 1. Now we're at anaphase 1. And anaphase is where homologous chromosomes pull apart. Spindle fibers are pulling them apart, right? And again, this shows in this case, this drawing is actually really quite confusing, right? Because this drawing would say that all the chromosome 1s are blue and all the chromosomes 2 are pink. Normally, you would actually have a pink one and a blue one heading in opposite directions at the same point. I've used this side before. I never noticed that. It's kind of cool. Any case. And then telophase is where the nuclear membranes start to reform. You start to get a cell, you start cytokinesis, which is why the cleavage furrow is showing, but your chromosomes start to relax. But none of this stuff has to go to completion because we're going to go do another cell division event. So sometimes telophase is not as complete here as it is in mitosis, but the same sorts of things are happening, and you certainly do get cytokinesis happening. So you do get 2 new daughter cells. Again, you start diploid, you go haploid. You start with 46 chromosomes. After S2, you go from 46 chromatids to 92 chromatids, although you still have 46 chromosomes. And after you finish your first cell division, you now have, in humans, 23 chromosomes in each cell, but each cell has 46 chromatids. Draw it out, make sure that makes sense to you. It's a good idea to try it with a different number. So pea plants have 7 chromosomes. Give it a try with 7 chromosomes and see if you can come up with the numbers. Then there's a really short phase called interkinesis, and interkinesis is basically a resting phase where the cells just get ready to do the next chromosomal division. There is no DNA replication here. There's no synthesis here. It's usually short. There's usually not a lot of growth here. They can do a little bit, but not very much. Then we go to meiosis 2, and this is essentially a mitotic division. So you started again with your two daughter cells, and you go to prophase 2, where all the normal prophasey stuff is happening. Then you line up on your metaphase plate, anaphase pulls them apart. Telophase, your chromosomes relax, your nuclear membrane reappears, your spindle fibers disintegrate, and then you do cytokinesis where you get to 4 haploid cells. What could go wrong? And this is the cool part, and we're going to talk about non-disjunction in a lot of detail later, but just the preview. If your pair of chromosomes doesn't separate, either in meiosis 1 or in meiosis 2, you end up in a situation where your gametes have one too many chromosomes or one too few chromosomes. And this is not the same as having an entire extra set of chromosomes. They're not haploid or triploid. They have it's called a trisomate. You have a third copy of a single chromosome, or a monosome. You have one copy of a single chromosome and an otherwise standard background. It turns out this becomes important in humans with things like Down syndrome. And in plants, they actually can be triploid, and things like bananas are triploid, and that's why you don't get big banana seeds, because when they try to do meiosis, they can't line up their chromosomes correctly because they're triploid, so they can't make seeds, and all the little black dots in your banana are aborted seeds. Bananas are all clones of each other, which has important implications for how they can fight off disease and that sort of thing.